Molecular basis of infectious disease Part 1. Feel free to pause the video to give yourself time to answer the questions. Mutations in which genes confer resistance to cytomegalovirus? UL97 and UL54. What does the viral gene UL97 encode? UL97 encodes a kinase that produces the triphosphate form of ganciclovir. What does the viral gene UL54 encode? UL54 encodes CMV-DNA-dependent DNA polymerase. CMV resistance mutations are more common in which gene? Resistance mutations are more common in UL97 than UL54. As opposed to UL54, the mutations in UL97 occur in hotspot locations. Resistance mutations in UL54 are more common under what treatment conditions? CMV resistance mutations in UL54 are more common with prolonged treatment of ganciclovir as well as pre-existing UL97 mutations. How does ganciclovir inhibit CMV replication? Ganciclovir disrupts chain elongation by its incorporation into viral DNA. What is the first-line treatment for ganciclovir-resistant CMV? Foscarnet. What is the target of the CMV antigenemia test? PP65, which is a structural protein that is expressed on the surface of infected polymorphonuclear lymphocytes. What is the inhibitory concentration 50 defined as? The concentration of an antiviral agent resulting in a 50% reduction in viral nucleic acid values compared to those of the control. In HPV infection, what does E6 bind to? E7 binds to P53, promoting its degradation. In HPV infection, what does E7 bind to? E7 binds to RB, resulting in the release of E2F and promoting the transition from G1 to S in the cell cycle. What is thought to be responsible for the perinuclear halo seen with HPV infection? The perinuclear halo is believed to result from accumulation of E4 protein. Is HPV DNA testing recommended for women younger than 30 years old? No. Pap testing alone is preferred for women younger than 30. There are 3 options for women ages 30 to 65. Option 1 is Pap and HPV co-testing every 5 years. Option 2 is a Pap test alone every 3 years and option 3 is HPV testing alone every 5 years. What are the steps in HPV hybridization capture? The key-argon hybrid capture for HPV is a method to amplify the signal using RNA probes and antibodies specific for RNA-DNA duplex hybrids. The first step is the denaturation of the sample. Second is the hybridization of the denatured sample with HPV RNA probes. Then hybrids are captured using monoclonal antibodies on a microtiter plate. A second monoclonal antibody conjugated to alkaline phosphatase is added. The alkaline phosphatase splits a chemiluminescence signal to produce light. This light is then measured using a luminometer in relative light units. I hope you enjoyed this video. To get updates when similar videos become available, please consider liking and subscribing.